These are the lecture notes for Chapter 15 on genes and proteins. Here is a list and structural chemical formulas for all the 20 different amino acids divided into categories. This group on the top left are nonpolar amino acids whose R group is nonpolar. The R groups are the groups in blue, and these are the groups that make each amino acid different from each other. So these amino acids that are nonpolar would be soluble in lipids and insoluble in water. And then we have a list of polar uncharged, which would be soluble in water. And we've got positively charged, negatively charged, and then nonpolar, again, aromatic. The first group was nonpolar aliphatic, which you don't need to worry with that. So we know that DNA codes for our traits, but the central dogma states that what DNA really does is DNA codes for messenger RNA, and that's called transcription. And then messenger RNA codes for proteins, and that is called translation. So transcription, translation. So you can also write it this way: DNA codes for messenger RNA, and then that messenger RNA takes the message from the nucleus to the cytoplasm to a ribosome, and that is where the protein is made. So transcription, this first part, happens in the nucleus. And then translation, the second part, where messenger RNA codes for a protein, this happens in the cytoplasm on a ribosome. So the flow of genetic information, the central dogma, is illustrated here. We can see that this is our original DNA, double-stranded DNA molecule, and one strand is used as a template to produce a single-stranded messenger RNA molecule. So RNA polymerase is the enzyme that just simply adds complementary nucleotides. For example, here the DNA had a C and the RNA polymerase added a G, and then the DNA had an A, the RNA polymerase added a U, C, RNA polymerase added a G. Remember, the RNA is going to have U in place of T, okay? So the messenger RNA is transcribed in the nucleus. It's then processed, and that's called RNA processing. And what happens there is there are pieces of the RNA that don't code for anything. So they are spliced out, and they are called introns. So the messenger RNA that leaves the nucleus is smaller in two ways. It is shorter and it's thinner because it's only a single strand, and it's shorter because some of the coding nucleotides are that they're non-coding. Sorry, they are spliced out. They're called introns, and the messenger RNA is left only with the pieces called exons, and there's going to be a a cap. I'm trying to think of the name of that. 7-methylguanosine, I think, is the name of that cap. You'll find out in your notes. And then a poly A tail, many A's. So many adenines form the tail. And after that, your messenger RNA has been processed, and then it's going to attach to a ribosome, and this is where translation is going to occur. So the arrow is pointing to the messenger RNA, and then what's going to happen is all these individual, the green, the yellow, and the blue, are transfer RNAs. They are tRNAs, transfer RNAs. So transfer RNAs only have 3 nucleotides, and they're going to be complementary to each codon, each group of 3 nucleotides on the messenger RNA. And so they're going to transfer the amino acids to the correct spot on in the protein, in the polypeptide chain, which will become the protein. Degeneracy is a property of the genetic code, and it means that basically there are 20 amino acids, and then there are 3 stop codons. And a codon is just a sequence of 3 nucleotides in messenger RNA. So the 3 nucleotide sequences in messenger RNA are known as codons. So for example, the first one is always AUG. That is always the start codon, and it codes for the amino acid methionine. So methionine is always the first amino acid in every protein. But 3 codons code for stop. That just means to stop making the protein. That means you're done, okay? So there would be 61 possible codons for just 21 amino acids. Sorry, for just 20 amino acids. That means that each amino acid has several different codons that can code for it. And here it describes it better. This is the codon chart. And you can see, let's just take one, phenylalanine, which is PHE. That's an R, which is PHE, phenylalanine. Okay, so phenylalanine can be UUU or UUC. And then let's see if we see it anywhere else. Nope. Okay, let's look at another one. Let's look at one that's got, let's just look at the next one, leucine. Leucine, Leu, has 6 possible codons. UUA, UUG, CUU, CUC, CUA, or CUG. Any of those codons code for leucine. And so if there is some kind of mutation, for example, if CUU, there's a mistake and that U becomes CUC, that's a point mutation that ends up being a silent mutation because it codes for leucine either way. So it does not change the amino acid and it doesn't change the protein at all. So that is degeneracy. Now we're going to look at a frame shift mutation. We've talked about these before, but we're going to see in the original, the U and the A are going to be deleted. So the G would be followed by CCC. Okay, so that's going to change the sequence of amino acids. Now, learning this information will help you to do your virtual lab that goes with this chapter. So it changes from serine, valine, proline, tyrosine to the last 3 amino acids being different, alanine, leucine, and leucine. And that changes the entire protein. Some changes are big and some changes are small. Some are bad and some are good. But it does change the protein when even one amino acid is different. All right, so let's start with the first stage, transcription, and that is when DNA codes for messenger RNA. And this happens in the nucleus of the cell. The first step is initiation, and that's going to be RNA polymerase binding to a promoter, which is a piece of DNA that kind of gets transcription started. Then there's the elongation, and RNA polymerase is going to add nucleotides that are complementary to the piece of DNA that's being transcribed. And then termination is when it stops. And this is a picture showing you initiation. It says the alpha, that's out yet. Is that alpha? The alpha subunit of prokaryotic RNA polymerase recognizes consensus sequences found in the promoter region upstream of the transcription start site. So this is where transcription is supposed to start, and there is a region that's minus 10 nucleotides down from 0, and then there's a region that's 35 nucleotides down from 0. So this one would be the 35th nucleotide. That's what -10 and -35 mean. So the RNA polymerase, the promoter actually includes both of these regions. The RNA polymerase binds to that promoter, and then it can start adding nucleotides at this point that are complementary to the DNA. This is a video that you can watch of transcription. It's slide number 10. And then elongation is pretty simple. It's very much like DNA replication, but you have to remember that the RNA that's produced, the messenger RNA molecule, everywhere that there was a T is going to be a U. So A's are going to be complementary to U's. Now T's are still A's. So the DNA contains a T, the RNA would contain an A. But if the DNA contains an A, then the RNA, the complementary nucleotide, would be a U. And termination occurs by interaction with a Rho protein and formation of a messenger RNA hairpin loop. There are two types of termination signals. They're Rho-dependent and Rho-independent. The Rho-dependent are controlled by the Rho protein, and the Rho-independent are controlled by sequences in the DNA strand. So, and this illustration just shows you that multiple polymerases can transcribe a single gene, while numerous ribosomes concurrently translate the messenger RNA transcripts into polypeptides. In this way, a specific protein can rapidly reach a high concentration in the bacterial cell. So the point is that many proteins can be made at one time. And this is transcription, the entire process. It's slide 14 if you would like to watch it. Now, these are the polymerases that are found in eukaryotic cells, cells that have a nucleus. There's RNA polymerase 1, which transcribes our ribosomal RNA genes. And those are, ribosomal RNA is a portion of the ribosome. Then there's RNA polymerase 2, which transcribes protein coding genes. And then there's RNA, and this is the RNA polymerase 2 is the one that we're looking at here. Okay, well, it could be any of them, but we were saying that this RNA polymerase is transcribing messenger RNA, which is going to then go into the cytoplasm and code for a protein. But that messenger RNA could also be coding for ribosomal RNA genes, or it could be coding for transfer RNA and small ribosomal RNA genes, and small nuclear RNA genes. So RNA polymerase 3 is the third type.